Um, Good morning. We talked about historical aspects of neurohumoral transmission. Now we'll talk about general considerations of autonomic nervous system. See, nervous system in our body are of two types. One is autonomic nervous system, another is somatic nervous system. Autonomic nervous system, we also call it vegetative nervous system or involuntary nervous system. Visceral nervous system, we also call it. And this is different from somatic nervous system in many ways. Somatic nervous system is voluntary nervous system. So it differs in many ways. Like autonomic nervous system, it innervates all organs except skeletal muscles. Skeletal muscles are innervated by somatic nervous system. Autonomic nervous system has ganglia. Somatic nervous system does not have them. And that is why we say that there is a synapse in periphery. This does not happen in somatic nervous system. The body of somatic nerve, the last synapse in somatic nervous system is in the cerebrospinal axis only. It forms plexuses also. Many of the visceral organs, esophageal, splanchnic, they are all plexuses. The nerves, they distribute to form a network like a net. Because of peripheral synapse, there is short postganglionic fibers compared to somatic nervous system that has very long postsynaptic fibers. So all these differences, there is difference in myelination also. Postganglionic fibers in ANS, they are non-myelinated, preganglionic are myelinated. So there are many differences between somatic and autonomic nervous system. Then we come to another important aspect, and that is sensory neurons, because an arc has to be completed. That may be, suppose it is a spinal reflex, this may be a short arc, or if CNS is also involved, then other longer arc will be prepared. But in an arc, important fibers are sensory fibers. So if we see autonomic nervous system, the sensory fibers are again of two types. One is parasympathetic, another is sympathetic. This is the basic division of autonomic nervous system: parasympathetic and sympathetic. So in sensory fibers also, we see that the parasympathetic sensory fibers are mainly through cranial nerves. Cranial nerves in sensory, most of the cranial nerves, we have to remember, they are mixed. They have both sensory as well as motor fibers. But some of them are prominent. Say sensory fibers of parasympathetic nervous system are mostly carried by 5th trigeminal, 7th, 9th, 10th, vagus, glossopharyngeal, trigeminal, and 7th is facial nerve. So these are the main cranial nerves which carry sensory fibers, and there is a sort of distribution also, like the areas from which they will carry, like trigeminal nerve. Trigeminal nerve, that we actually study more in embryology. Trigeminal nerve, it carries the sensations from the deeper parts of the face and head. This is the area that is covered by trigeminal nerve. Tongue by 7th nerve, facial nerve. If you see hard palate and upper part of oropharynx, this is from 9th, and lower part of oropharynx to whole organs, visceral organs, that is through vagus nerve. So these are the sensory fibers which carry, and remember in parasympathetic also, like in efferent also, there is sacral element also. Sacral, second, third, fourth sacral, they carry sensory fibers also. So this efferent fibers, I can say, or sensory fibers of parasympathetic are through these cranial nerves. We can remember some element is there in through sacral nerves also. On the other hand, sympathetic, just like motor fibers or efferent fibers, all thoracolumbar sympathetic nerves, they are mixed. They carry efferents also and efferents also. We have to remember one more thing, that even though we rely more on synaptic reflex, but in parasympathetic system, most of the activities are coordinated at central level. Actually, solitary tract nucleus, STN, along with hypothalamus, is the major area where we can say all these autonomic activities are regulated. So this we have, though other centers are also involved right up to cortex, but STN and hypothalamus are the major organs which regulate the activity of autonomic nervous system. I found a very good diagram in Goodman-Gilman, and I thought this also is from Goodman-Gilman. This shows the difference. I will take another one. So this is the difference between various types of nerves. Why I have taken them specifically from the book? Because we can understand everything through these diagrams. They are very nice and illustrated diagrams. So see, this is the motor neuron of somatic nerves. So here we can see in the spinal cord the body is there, and it directly goes and goes to skeletal muscles. There is no ganglion in between. So this is a long fiber. Compared to this is autonomic nervous system. This is parasympathetic, cranial and spinal. This is going to the ganglion. This is preganglionic fiber. This synapses in the ganglion. And postganglionic fiber is very small. That is now innervating the smooth muscles, cardiac tissue, secretory glands. This is sympathetic. Here thoracic and lumbar. Comparatively, you will find in sympathetic system the postganglionic fibers are longer. This is the ganglion, and some of the fibers, they go directly to adrenal medulla. Presynaptic fibers, they go to adrenal medulla, and from there is no postganglionic fibers. There is release of epinephrine. So this is also, and from the ganglion, postganglionic fibers, they go to smooth muscles everywhere, and it is also seen that sympathetic cholinergic fibers, we have to remember them specifically. Some sympathetic cholinergic fibers also are there, like in sweat glands. We'll talk about them. So in this diagram we can understand the differences in the innervation by autonomic and then other diagrams. One diagram is very comprehensive. See this. This diagram has all elements. This shows the sensory, though he has only shown through 9th and 10th. I told you trigeminal nerve and 7th facial nerves are also involved in sensory input. But we have to understand this diagram very nicely. Some of the lines are dotted, some of the lines are continuous. These continuous lines are depicting presynaptic fibers and dotted lines are depicting postsynaptic. Yellow means cholinergic, red means adrenergic. This is aberration like in blue color, that is sensory. So here we see, first of all, if we talk of sympathetic nervous system. Now I am talking about efferents, motor, efferents. Sympathetic nervous system, like we had seen in that diagram, it has ganglia very near to cerebrospinal axis, spinal axis. There are 3-4 types of ganglia we can see. One are paravertebral, in the sides of the spinal cord. Another are prevertebral. Prevertebral are 3-4 types. There are 3 types shown here, like celiac ganglion, superior mesenteric, inferior mesenteric. There is orticorenal ganglion also, which are seen near the kidney. And third are terminal. There are some terminal ganglia like in near the rectum or near the kidneys. They are very near to the organ. So there are these 4 types of ganglia, sympathetic ganglia, which are seen. So we'll talk about them. These are paravertebral ganglia. 22 pairs of paravertebral ganglia are seen, and they receive preganglionic fibers from T1 to L2 to 3. First thoracic to second or third lumbar segments of spinal cord, they hold the cell bodies of these preganglionic fibers. So they ultimately synapse in these ganglia. So let us talk of paravertebral ganglia. I told you 22 pairs. This is how what is shown here is intermediolateral part of the spinal cord. This is the cell body of preganglionic fibers for the sympathetic. Through this ventral root, this is ventral, this is dorsal. So what is shown here is sensory. They are coming through dorsal, but this efferents from the spinal cord, they are going to, this is the paravertebral ganglion. So through ventral root, these fibers, yellow color means cholinergic fibers, and remember preganglionic fibers are myelinated. They are myelinated. So they go to the paravertebral ganglion, and on this side you can see here, this is white ramus we call it, because they are myelinated and it looks white. This nerve looks white. So coming out through ventral root or anterior also we call it, ventral is anterior, and then going to ganglion through this, which we say white ramus communicans. White because it is myelinated preganglionic, and from this ganglion, postganglionic fibers, they again go to the nerve, vertebral nerve, for distribution to hair follicles, to blood vessels of skeletal muscles. So this, now this is postganglionic. Postganglionic, I told you, are non-myelinated, so we call it gray ramus. So these 2 rami you will find near the paravertebral ganglion. One is white, that means it is carrying preganglionic fibers, and another is gray, that is taking postganglionic fibers. So postganglionic fibers in the area, it is like for the trunk, for limbs, they are distributed through this vertebral nerve only. It is not the case everywhere. Remember the visceral organs. There is no visceral supply to vertebral nerves. Vertebral nerves, they carry to the trunk and limbs. So the fibers which are to supply the trunk and limbs, they are going through gray rami communicantis to the vertebral nerve only. Rest of them, they are going directly to the organs. So this we have to remember. So we can see here that this distribution is only from first to second or third of lumbar, but there are cervical ganglia also. So we have to remember that it is not a rule that the fibers which are originating from T1 will go to paravertebral, this ganglia only, which is in the vicinity. These fibers, they originate from here and they can come down, they can go up. That is how there is cervical ganglion also, and we can see distribution up to iris, up to eyes. But these fibers are originating in thoracic area only. So thoracolumbar, but if we see distribution, it is from top, from the eyes to limbs also. So these fibers, it is not a rule that they will come out and immediately synapse in the nearby ganglion. They can travel down also, they can travel up also, 3, 4, 5 ganglion down. So we have to remember that. Then we come to prevertebral ganglia. As I told you, there are 4, but mainly we study about 3 of them. Celiac ganglion, this roughly they start from L5 to T12. These fibers, they come out and they do not synapse in the paravertebral. They travel and then they come to celiac ganglion. Similarly, some of these fibers, they form superior mesenteric and inferior mesenteric ganglion. So these ganglia are situated very near to the organs comparative to paravertebral ganglia. And some of those, these celiac ganglion, this innervates these organs, and superior mesenteric ganglion, we can see distal colon, and inferior mesenteric, urinary bladder, external genitalia. So they synapse at this level. Terminal ganglia, I told you, very few. Some of them are near kidney, some of them are near rectum. It has been observed. Along with this, plexuses are also formed. As I told you, esophageal, cardiac plexus. So splanchnic, we all know. And remember, there is a full-fledged enteric nervous system. So these fibers, they will go and innervate enteric nervous system. We'll come to that. So this is sympathetic supply, efferents. If we see parasympathetic, just like I said, these are the nerves which are involved in the efferents of parasympathetic. 3rd, 7th, 9th, 10th, and sacral, 2, 3, 4. So we call it craniosacral. Actually, sometimes we don't even call them sympathetic and parasympathetic. We just say craniosacral outflow and thoracolumbar outflow. So thoracolumbar is sympathetic, craniosacral is parasympathetic. So these nerves, if you see 3rd, and actually the bodies of craniosacral is situated in CNS, in the brain. We say from midbrain to medulla oblongata. This is an exception, sacral. Most of the parasympathetic outflow is from midbrain to medulla oblongata. Midbrain is characterized by this 3rd nerve, Edinger-Westphal nucleus. So this is situated in midbrain. We call it tactile outflow. So this will come out through 3rd ciliary nerve and will synapse in ciliary ganglion. Ciliary ganglion will innervate the iris muscle, ciliary muscle. And then through this seventh has two elements. Facial nerve has two elements, two parasympathetic outflows. One is through this chorda tympani. Chorda tympani will innervate sublingual and submaxillary. There are ganglia which are situated very near to this submaxillary area and sublingual area. So this will, chorda tympani will go and synapse there. Another is this petrosal nerve, superficial petrosal nerve. This goes and synapse in sphenopalatine ganglion. That innervates lacrimal glands. So facial nerve has these two branches. And then we come to ninth. This synapse in otic ganglion, that we can see innervates to areas parotid gland. And then comes to this. Remember these 3rd, 7th, 10th, they will cover mostly the upper parts, head, mucous glands of mouth, nose, salivary glands, lacrimal glands, all of them we can see are covered till 9th. 10th is very unique. Takes everything, the whole body except the pelvis. Pelvis is innervated by sacral outflow. Otherwise, vagus nerve is innervating all organs of our body. And we can see here that this is traveling full. This is forming part, this is forming various plexuses, like we can see vagus going to heart directly. And this is a continuous line, means preganglionic. So this is basically forming a plexus, cardiac plexus. This is synapsing there. It is coming near the liver again, hepatic plexus. It is coming down near, we can see spleen, kidney, intestine. So these plexuses are formed. So vagus nerve innervate all visceral organs except, as I said, pelvic organs which are innervated by sacral outflow. Otherwise, vagus nerve is responsible for all these activities. Then we can see postganglionic fibers here, which are seen, we'll talk about that, but remember, all preganglionic fibers are cholinergic. All preganglionic fibers are cholinergic. Yesterday also we were talking that acetylcholine. All preganglionic fibers. And some of the postganglionic fibers, like here you can see sweat gland and specialized blood vessels of lower limb. So these fibers are coming from the sympathetic ganglion, but they are dotted yellow in color. So they are specialized fibers. They are also cholinergic. All parasympathetic pre and post, all are cholinergic. But except these exceptions, postganglionic sympathetic fibers are adrenergic. We can see here dotted lines. We have to remember one more thing, as I've already said, in sympathetic there is no efferent above first thoracic. So they are going up and forming celiac ganglion. These three celiac ganglion we can say, superior, middle and inferior, and there are stellate ganglion also. So what they do, they innervate the, you can see here from superior cervical ganglion, the postganglionic sympathetic fibers are going to eyeball, supplying the iris, the ciliary muscle. They are going to sublingual, submaxillary. So these are cervical, they are supplying the head and neck. So we have to remember this. So this, I found this diagram is very comprehensive. If we understand it nicely, it becomes very easy to revise. Then after this, we come to how this conduction takes place. As we know, in the nerve, the conduction is electrical through voltage gated sodium channels. They are sensitive to the voltage in the vicinity. So when depolarization takes place at some point, then the voltage gated sodium channels in the vicinity recognize it, and then we say that when they recognize it, they open. So the negativity inside the cell is converted into see this. Normally there is negativity inside the cell. When the depolarization takes place, action potential we call it, then it shoots to some positive. Sodium positively charged ions will enter and this will become positive. This is what we say depolarization. Now this depolarization needs to be propagated. So the voltage gated sodium channels in the vicinity will recognize this depolarization and will open. And this is how this will travel. This will travel up to the synapse. What happens at the synaptic level? At synaptic level, it is shown here that this nerve comes and synapses. Two types are shown here. One is shown as excitatory, another is inhibitory. So in excitatory, we can see sodium channels in the postsynaptic nerve, or it may be muscle also. They open, sodium will enter and will depolarize this postsynaptic. And now this depolarization will also be propagated. So this is how normally happens in excitatory. In inhibitory, the channels which open at the postsynaptic level, they are inhibitory, like chloride, potassium. Remember, potassium is positively charged, but the concentration inside the cell is very high. So whenever potassium channels will open, potassium will come out. When potassium comes out, then the cell, we say, gets hyperpolarized. Or if the channels for negatively charged ions like chloride, they open, then chloride will enter inside and will inhibit. There will be no depolarization of postsynaptic nerve or muscle. So that is shown here. This is, we say, IPSP, inhibitory postsynaptic potential. And here it is shown excitatory postsynaptic potential. So excitatory means in postsynaptic also the wave front will move, it will be propagated. In inhibitory postsynaptic, this stops here only. There will be nothing in postsynaptic junction. So these two types. Now how this takes place, that is very important. How this communication from presynaptic to postsynaptic, or from postsynaptic to the effector organ, it takes place. Before that, we should also understand what are the functions of ANS. We all know about it, but just for the sake of remember, we can say that sympathetic nervous system is not required. If I don't have extremes, if I lead a comfortable life, you don't need sympathetic nervous system, means it is compatible to live without sympathetic nervous system. Sympathetic nervous system is more diffused and it is responsible, what we say, fight or flight. So in emergency conditions, you need sympathetic, where you need more blood supply, where you need more glucose. These are the functions. Remember, the functions of autonomic nervous system are related to respiration, blood supply, cardiovascular, I can say, reproductive organs, blood volume, emotions, sleep pattern, and this is what we say homeostasis. So in One word if you say, then autonomic nervous system is responsible for maintaining homeostasis. But sympathetic, as I said, is more diffuse. One of the reasons for that is also adrenal gland that secretes epinephrine. This has more generalized effect, like if blood pressure increases, this will increase in the whole body. On the other hand, parasympathetic is more localized, and we can say that it may not be compatible to live without parasympathetic nervous. So this is more important for us because this is involved in what we say rest and reproduce. For getting the nutrients, you need parasympathetic system. For reproduction, you need parasympathetic system. So parasympathetic system is more important to our body. But as I said earlier, if you cut the autonomic nervous system fibers, the organ will keep working. Most of the cells, or almost all of the cells which are innervated by autonomic nervous system, they have their inherent activity. So they will keep working. And there are some localized, as I told you, nerve plexuses also are there. Like we talk about enteric nervous system. So even if we cut vagus nerve, the GIT will keep working because of enteric nervous system. So some autonomy is there with our visceral organs. So these autonomic nerve fibers, we can say, they just regulate the activity. They do not stop the organs if they stop working. This is unlike what happens in somatic nerves. If we cut the nerve, the muscle is paralyzed. Here the organ is not paralyzed. So that also we have to understand. So this very important. We'll talk about that in next one. Synaptobrevin, synaptotagmin, and some RAB also. In the presynaptic and postsynaptic SNAP25, MUNC18, MUNC, I call it MUNC, easier to remember, and syntaxin. Just remember them, we'll talk about them. So we take this first. This is what? These are synaptic vesicles. Remember, we call it neurohumoral transmission. Means some chemical is involved in this transmission from presynaptic to postsynaptic, and from postsynaptic to effector. This is not electrical. This is chemical transmission, and that is very important. What we actually means by neurohumoral transmission is we are talking about this chemical transmission also. We call it neurohumoral. There are various reasons. Remember, autonomic nervous system rarely you will find is independent at central nervous system level. We cannot actually differentiate that this part will manage only autonomic nervous system activity. Somatic part is always there. That is why actually when we have some activity, this is mixed. When the skeletal muscles will work, autonomic nervous system will be stimulated. So they are all mixed together. There is no exact division in the CNS. All are connected, and actually even hormonal system is connected. When some CNS activity is going on, that controls our body through pituitary. Pituitary gland will also try to modulate the things. So all these things are interconnected. Here it is a chemical transmission. In the history we had seen how it evolved, but now we know a lot about it. When we say that this is the neurotransmitter, we have read it in schools also, that how to prove that this is the neurotransmitter at this synapse. Then we say this neurotransmitter should be stored in the vesicles, in the presynaptic nerve. We say there should be presence of enzymes which is causing the synthesis of this. Then we say on stimulation of presynaptic nerve, this neurotransmitter should be released. Then we say there should be some receptor at the postsynaptic level for this chemical, then only we will call it neurotransmitter. Then we say there must be some mechanism to terminate this effect. Then we say there must be some mechanism to block the activity of this neurotransmitter. So all these are criterias which are required nowadays. As I told you, full-fledged discipline is there. So one of the important criteria is this neurotransmitter should be stored, should be synthesized, and should be released. Now this neurotransmitter is stored in the vesicles, in presynaptic nerves. And these vesicles, when the depolarization front comes at the nerve terminal, then these vesicles, they are combined with the membrane, nerve terminal, and this releases the contents. It's not only the neurotransmitter, other contents also they are released, like ATP and some other. They are also released in the synaptic cleft. And this we have studied in detail now. We should have some knowledge about that. This is the process. If you see, this is the synaptic vesicle. Now this will do what? This is the synaptic vesicle. When the stimulation of presynaptic nerve takes place, then this comes in the close proximity of this neuronal membrane, nerve terminal. This we call active zone. This process is known as docking. When the vesicles, they are in the cytoplasm of this nerve, and remember, the cell body is not situated in the terminal. We have to remember this. So most of the enzymes which are synthesizing the neurotransmitter, they are synthesized in the body of the neuron, and then they travel and reach the nerve terminal where the vesicle is there. This also we have to remember. So this is known as docking. Then this vesicle gets activated for releasing the neurotransmitter, that we call priming. And when this fusion takes place with the neuronal membrane, the contents are released, then this can be reused. Either immediately this can be filled with neurotransmitter, or this can become completely empty and can become a part of early endosome, means starting from the beginning. This will become a part of the lysosomes. There are those sheets are there. So endosomes, early endosomes. And then the whole process starts again. We call it budding. Budding. If you remember those sheets, then those vesicles are broken, empty vesicles, Golgi complex. If you remember, Golgi complex, there are like sheets, and then these vesicles, they break away from those sheets. They are empty vesicles, then they are filled with required enzymes. So this can be another process. So any of these mechanisms can be followed by this synaptic vesicle. So how this is stimulated, how this activities are taking place. See this. This is docking, active zone. This is synaptic vesicle. Remember at least these two these two parts. One is synaptotagmin. Remember, we call them snare proteins. Snare means one is N-ethyl maleamide sensitive factor, NSF, and then NSF activated protein, SNAP we call it. SNAP, S-N-A-P, capital. It's an acronym. NSF activated protein, SNAP. And snare means SNAP receptors. So it becomes snare. So remember these NSF, N-ethylmaleamide, sensitive factor, that is NSF, and NSF activated protein is SNAP, and SNAP receptors are snare. So all these are known as snare proteins. Snare. So these are in the synaptic vesicles. What we see are synaptotagmin and synaptobrevin, very important. And on the neuronal membrane, three, four of them, complexin is also there. One is SNAP25, another is syntaxin 1, and this is Muncy 18. So we have to remember them. What happens in priming? Priming also, priming 1 and priming 2. In priming 1 we say that this Muncy 18, MUNC 18, it binds with syntaxin 1 and stabilizes it. Actually we can see here even in docking, this Muncy 18, this binds with syntaxin 1 and this stabilizes it for binding with SNAP25 and vesicle. So in priming, what is happening? We can see this is SNAP in black color, this is SNAP25. This is syntaxin 1 that was stabilized by this Muncy 18 in purple color. So all of them, they come together and forms a complex with synaptobrevin, this in green color. Synaptobrevin was with this synaptic vesicle. So all of them, they come and form a complex, they bind with each other. So now synaptic vesicle is attached to the neuronal membrane. Then in priming 2, this complexin in blue color, this comes and attaches to this SNAP25 and syntaxin 1. This is important. And what is synaptotagmin in red color? This is the protein that binds with calcium. Remember, when we say stimulation, then we are talking about opening of calcium channels. When the depolarization front reaches the nerve terminal, then this opens calcium channels. These calcium channels, they enter at the nerve terminal and they bind with this synaptotagmin. This synaptotagmin calcium complex in red color, this binds with this complexin. This red color is synaptotagmin along with calcium. Calcium has entered and this binds with this complexin. Complexin actually helps in binding of the synaptotagmin calcium complex to this complex that is formed during priming 1. And when this happens here, this is fusion pore opening. Now this part of this synaptic vesicle membrane will fuse with neuronal membrane. Open all the contents of these synaptic vesicles, they will be released in the cleft. And then this will again separate, return to ground state like we had said earlier. Either this can again take up the neurotransmitters directly, and remember this entry of neurotransmitter in these vesicles is dependent on hydrogen ions. ATPase is there that produces actually sodium potassium ATPase is there. Sodium hydrogen pump is there. So that causes concentration of like protons like we see in gastric mucosa. So protons are concentrated in the synaptic vesicle and these protons are then exchanged with the neurotransmitter. Remember, the process of synthesis of neurotransmitter starts in cytoplasm. The precursors, like if suppose we'll talk about that in cholinergic transmission, if it is synthesis of acetylcholine, the choline is coming from outside. So then this has to be taken, combined with acetyl coenzyme A, and then it is taken in the vesicle. So this exchange takes place because of exchange with protons, hydrogen ions. So these hydrogen ions, they are stored in these vesicles and then they are exchanged with these neurotransmitters. So either immediately this neurotransmitter can enter inside this vesicle, or like I said, it can go and directly meet with the endosomes, become a part of endosomes, and then fresh vesicle will come out and this will keep recirculating. So this is how this transmission in the synapses, either in ganglion or in effector junctions, it takes place. Now from next time we'll start specifically with cholinergic and we'll see how that release takes place.